In this video, we're going to look at two related metabolic pathways that take place in the mitochondria of eukaryotic cells, and these are going to be pyruvate oxidation and the tricarboxylic acid cycle. Well, remember that cells have a number of different metabolic pathways. They can carry out glycolysis to produce pyruvate, and then the focus of this video will be what happens if there's oxygen present in the cell and they're able to import the pyruvate into the mitochondrion and use the TCA cycle. Well, let's begin by orienting you. This is a cartoon of a mitochondrion, and here I'll add some labels so you can see where the cytoplasm is, the outer mitochondrial membrane, inner membrane space, inner mitochondrial membrane, and matrix all are. Pyruvate is produced in the cytoplasm using glycolysis. It's a 3-carbon compound. It's quite small, and what that means is it's able to freely pass through the porins in the outer mitochondrial membrane and enter the intermembrane space. But the inner mitochondrial membrane is not very permeable, and so in order to move from the intermembrane space into the matrix, a transporter is required. We call this the pyruvate transporter, and it will import pyruvate into the matrix. All right, well, let's blow this up so we can see what's happening in the mitochondrion. Pyruvate's the starting material for these two reactions that I'm going to talk about. The first, called pyruvate oxidation, will convert pyruvate into a compound called acetyl coenzyme A. And then the second, the tricarboxylic acid cycle, which is also called the citric acid cycle and the Krebs cycle, will further metabolize that. Let's look at them one by one, beginning with pyruvate oxidation. So here's our starting material, pyruvate. What happens in pyruvate oxidation is the pyruvate's combined with a molecule called coenzyme A. This produces acetyl coenzyme A, and also one of the carbons in pyruvate is released as carbon dioxide. This reaction is catalyzed by the enzyme pyruvate dehydrogenase, and that name should make you suspect that it's a redox reaction, and it is. It's coupled to the reduction of NAD+ to form NADH. Now, coenzyme A is a pretty gigantic molecule, as is acetyl coenzyme A, and it's not really important that you learn their structure. I do want to show you, though, that the two carbons from pyruvate are going to be attached to this end here where there's a sulfhydryl group. So if you compare the structures of acetyl coenzyme A and coenzyme A, you can see where the two carbons have been added. Now, this reaction is considered an energy-yielding reaction in that it yields NADH. NADH, the reduced form of NAD+, has two high-energy electrons that will be able to serve as a starting material in a later metabolic reaction to get out ATP. Let's set that aside for now and instead look at the second set of reactions, the TCA cycle. Well, here the starting material is considered acetyl coenzyme A, and what will happen in that first reaction is the two carbons on acetyl coenzyme A will be combined with a 4-carbon compound called oxaloacetate in order to create the 6-carbon citrate molecule. This reaction's synthesized by the enzyme citrate synthase. To keep things simple, I've chosen not to show you the chemical structures of most of the compounds in these reactions because I think it can be complicating, but I'm going to make an exception for citrate. Here it is. And although I flipped it on the side relative to the cartoon version with the balls, I hope that you can see the 6 carbons, the 5 carbons in a row with the 6th carbon attached to the middle carbon. Citrate is the deprotonated form of citric acid, and in the cell, citrate and citric acid are converting back and forth between each other. So when we say citrate, we're really talking about a form of citric acid, and that explains why the TCA cycle is also called the citric acid cycle. There's something else I want you to notice about citric acid, and that's that it has 3 carboxylic acids, COOH groups on it. That explains why it's also called the tricarboxylic acid cycle. And since I'm talking about the names, let me just say that the Krebs cycle, that comes from the name of the biochemist who worked out many of these details. All right, well, let's make some more room and move on to the reaction itself. In the first step, citrate is converted into isocitrate. The isocitrate then is converted into alpha-ketoglutarate, and what I want you to notice is that while isocitrate's a 6-carbon compound, alpha-ketoglutarate is only 5 carbons. The 6th carbon is released as a carbon dioxide in this reaction. This reaction is catalyzed by the enzyme isocitrate dehydrogenase, and that's a clue that it's going to be a redox reaction. And in fact, this reaction also reduces NAD+, forming a molecule of NADH. The next step is complicated. Alpha-ketoglutarate is combined with coenzyme A to produce a molecule called succinyl coenzyme A. In this process, a carbon is released. Now once again, the enzyme name is a dehydrogenase, and so you won't be surprised to learn that there's another reduction event where NAD+ is reduced to form NADH. In the next step, the enzyme succinyl coenzyme synthase clips off the coenzyme A to generate succinate, a 4-carbon compound. This reaction has sufficient energy that it's able to simultaneously catalyze the phosphorylation of GDP to form a molecule of GTP, so it's an energy-generating step. Succinate is converted to fumarate by succinate dehydrogenase. Now this is another redox reaction, but in this case, the electrons from succinate are being transferred to a molecule of flavin adenine dinucleotide to form the reduced version of that molecule, FADH2. Like NADH, FADH2 will have high energy electrons, which can be used for the starting material in the electron transport chain. Fumarate is then converted into malate, and in the last step in this cycle, the enzyme malate dehydrogenase converts malate into oxaloacetate and at the same time reduces one more molecule of NAD+ to form NADH. What this means is that the overall yield for this reaction for each molecule of acetyl coenzyme A is 3 NADH molecules, 1 FADH2 molecule, and 1 GTP. Let's take a step back and think about the bigger picture. Each molecule of pyruvate then will be broken down through pyruvate oxidation, and this will generate 1 ADH, and then it will be further processed to generate these other products through the TCA cycle. Remember that pyruvate was produced by glycolysis in the cytoplasm, but the pyruvate oxidation in the TCA cycle will take place in the matrix of the mitochondrion. I'm going to wrap up this video by talking about what happens to the products generated through these reactions. Well, one of the things that gets produced is CO2. The 3 carbons in pyruvate are released as carbon dioxide. Carbon dioxide is a gas. It's very membrane permeable. It will pass out of the mitochondrion and out of the cell. The second product is GTP. And GTP is a bigger molecule. Some of it can be consumed in the matrix, but other GTP molecules will be exported to the cytoplasm. This is accomplished using an exchanger that exchanges one molecule of GTP for a molecule of GDP. And that's going to ensure that the raw material of GDP is present in the matrix where it's needed to run the TCA cycle, as well as providing GTP for the cell. The last products of the TCA cycle and pyruvate oxidation are NADH and FADH2. Those are generated in the matrix, and they will be consumed in the mitochondrion in a series of reactions that are going to be located in the inner mitochondrial membrane, and those are collectively going to be called the electron transport chain. We'll look at that in another video.